Thank you. And now I want to introduce Kevin Esvelt. He is a MIT professor who leads the Sculpting Evolution Group in exploring evolutionary and ecological engineering. He is the creator of a synthetic ecosystem to rapidly evolve molecular tools, and he's best known for inventing a CRISPR-based gene drive system capable of single-handedly editing wild species. He is the leader of efforts to ensure that all research in the field is open and community guided. His laboratory broadly focuses on developing new molecular tools and safeguarding biotechnology from mistrust and misuse. And now he will give a talk about sculpting the evolution of biology and technology. So welcome, Kevin. The stage is yours.
All right. Well, thank you so much for the opportunity to talk about something a little bit different than I normally do. I'm going to try to weave together a number of the themes that we've heard about so far today, from the philosophy to the science fiction to the actual science. And admittedly, as you heard, I tend to focus a bit more on engineering ecosystems using evolutionary engineering. But my lab is called Sculpting Evolution because we study the evolutionary process, which doesn't just apply to biology. It also applies to ideas, to culture, to technology. So what we're interested in is this question of, can we sculpt the evolution of technology to discover those that we think will be beneficial and stay away from those that could threaten everything that we've built? So most of the time I end up talking about engineering populations of wild organisms. So we came up with a way to use CRISPR, the genome editing tool, to encode it in the cells of organisms in such a way that the alteration that goes along with it will always be inherited, because the editing just happens every generation, and so it can spread through a population, which is great if you want to say get rid of malaria or otherwise edit wild species. But what's a little bit disturbing about it is that when I first thought of this back in 2013, no one had ever imagined that that might be possible, that a single researcher might engineer an organism in the lab that could, if released, eventually spread to affect the bulk of a species. So the point is just not everything is actually imagined and portrayed in science fiction before discovery. But most things are. So a book I read quite some time ago, so Heart of the Comet by Brennan Benford, gets into a lot of these questions of what would we engineer humans to be like in the future? And the obvious one, particularly obvious in the context of the last year, is viruses. So if anyone currently listening actually likes viruses, please write it in the chat. There's usually one or two virologists in the audience, or just someone who kind of likes the idea. But frankly, most people really aren't fans. And in science fiction, including this book, there's a common trope that future humans will be engineered to be resistant to viruses, or at least when they catch a cold, they don't get the drippy nose and the sneezes and the coughs and all of the miserable symptoms anymore, because those responses are snipped out. At least that's how it's described in Heart of the Comet. But what my group is interested in is, what can we use with CRISPR? Can we use CRISPR, which is really a bacterial immune system, against bacterial viruses, among other parasitic DNA elements? Can we repurpose that in mammals? So CRISPR is a system for recognizing snippets of viral DNA. So if you imagine all these are viral DNAs, if we can then design instructions to target CRISPR to each of these. So these are Cas12 guide RNAs that direct the Cas12 CRISPR system to each of the targets. So Cas12 binds each of these, then it takes this single guide RNA molecule and it separates it out into the different elements that target each of these three. And then the Cas12 enzyme uses them to find its target anywhere in the relevant cell, and then it can be cut. So this is how bacteria defend themselves against viruses. But as a commenter pointed out, there's an awful lot of viruses in the world. In fact, estimates range for just in mammals, somewhere between 40,000 and 3.2 million, is the range of the estimates. So the notion that we're going to predict which one is going to cause a pandemic next seems a little bit foolhardy. But that's not really the point. There aren't that many of them in humans. And the difference between a natural CRISPR system, which acquires a fragment of a virus to recognize it one at a time, and what we can do with engineering is we can program CRISPR systems to target many sites in a virus at once, which makes it much harder for it to evolve resistance. The problem is, in order to do this, we need to be able to target CRISPR to hundreds of different viral sequences. And the recognition signals that Cas12 uses to find its guide RNAs is just too repetitive if you try to stack up several dozen of them in a series. So what we did recently is we ran a bunch of experiments to analyze this scaffold. We made lots of mutant variants, and we checked to see, could they still direct Cas12 to bind a corresponding DNA sequence? And could they still mediate that processing step of separating out the long instruction set into each individual target sequence? And we found hundreds of variants that appear to be active. And so we can stitch these together into, so far, dozens, and we believe hopefully hundreds of different targeting instructions for Cas12. And initially, of course, we're going to try this in mice, because we try everything in mice first, in mammals. Programming the mice to be immune to entire families of mouse viruses that we know about. All of the mouse parvoviruses, for example, all of the mouse pox viruses, we want to engineer them to be completely immune to. Maybe it will work, maybe it won't. But if so, this is the sort of thing that we could plausibly, eventually, engineer into people to provide programmable virus resistance. But in order to do that, we either need to engineer it into future people or find some way of editing enough of the cells of each one of us listening today. And the challenge is we need to get it into most of our cells. And that's hard because evolution has worked for quite a long time to make us resistant to infection by external parasitic DNAs, that is viruses. So when we try to get DNA into a cell to, say, encode it to be virus resistant, the immune system fights us. So I think what we need to work on is ways of engineering our cells to be engineerable, putting on DNA transporters in each of our cells that will recognize a particular unique molecular signature on an incoming DNA packet that then will be internalized and imported into the nucleus and stitched into the genome reliably. But we need to engineer the lock so that the key is something that can only be accessed through synthetic chemistry and not through biology. And that's because whatever we can program viruses, whatever we can program to resist current viruses can be evaded by designing the viruses. But now to this question of future people for whom it's much, much, much easier. We don't necessarily just want to put in virus resistance, which would be one packet of DNA, albeit a reasonably large one, but still one insertion. Should we do that? That's a very complex ethical question, but I would submit that I would be annoyed if my parents could have done that for me and didn't, because I would be basically the same person, minus the misery of getting infected by nasty viruses periodically. Wouldn't have helped me against SARS-CoV-2, of course, because that wouldn't have been around, but still better something than nothing. But what about more complex traits? That's what most people tend to care about. And I don't want to dive into the really controversial weeds here, but just point out, how could one do it? And right now, we have in vitro fertilization to make a number of embryos, and then pre-implantation genetic diagnosis, which lets you grow up the embryos in a dish, pick one cell off, and sequence the whole genome for each of the embryos, and then decide, based on those genome sequences, based on the predicted health profile, which one you want to implant. That's available today. So with that, you can plausibly enhance multigenic traits, things that are impacted by several dozen or hundreds or even thousands of alleles, but you only get a limited selective power because there's only a few embryos. And of course, making multiple embryos is controversial, but most human pregnancies don't make it to term. So I'm going to set aside the question of the moral status of embryos. I personally tend towards a approach of if it can't feel or suffer, then it's not morally relevant yet, but reasonable folks may differ on that front. What people are working on now in mice with hopes of applying to cows is something called iterative embryo selection. And the idea is, instead of just making embryos from current sperm and eggs, instead you use sperm and eggs from two parents and combine them in normal IVF. You make embryos, and then you use those embryos, you grab out embryonic stem cells from them, and you culture them in a dish, directing them to differentiate into sperm and eggs. And then, of course, you can sequence those embryonic stem cells first to figure out which genomes they have, and then you know which sperm and eggs to combine, which then, of course, gives you another embryo, or rather a whole lot of embryos, all of which you sequence again, check to see which ones have the alleles you want, differentiate again into sperm and eggs, and then combine, shuffle, repeat. So you're doing whole generations essentially in the petri dish until eventually you get the genome you want, or at least you're satisfied enough with. And then you implant that embryo, or in this case, in the cows, they plan to use cloning. So that's one way. If we can figure out how to do that, that would make it tremendously more powerful and give us control over much of the genome. But the other way is pretty compelling. And this is just the Carlson curve cost chart for DNA sequencing and then oligonucleotide, so short DNA fragment synthesis, and whole gene synthesis. And you'll note that the green line here is whole gene synthesis. And it's currently decreasing at a cost at a rate considerably faster than Moore's law. So one can project ahead and say, in about 20 years, the cost of making a whole human genome from scratch may well be, based on those projections, about the same as it will cost to attend MIT. So you can think about that for your future children, what you would prefer. Give them whatever advantages can be accrued through this kind of engineering, or set aside funds to send them to elite universities someday. You Europeans have it easy. The caveat for both of these methods is that we don't know how to reliably reprogram the marks that control gene expression in cells, that say which kind of tissue are you, which kind of cell. And that is why most attempts to clone mammals fail. And it's why Dolly the sheep was pretty unhealthy throughout much of her life and died young. So unless we can work those out, we obviously can't make future humans using either of these methods. And we'll be stuck with current approaches. So it's highly uncertain whether biology will really be able to reshape the genomes of future humans at many, many different sites throughout the genome. In principle, it's possible, but there's a lot of major barriers that remain. Still, supposing that we could do it, even to a limited extent possible today, what are the most important complex traits to optimize? Again, reasonable people may differ, but I submit that they are morality and wisdom. And that's because if humanity is technology, then which technologies we use determine what humanity is like. Not just the future people, but also the way we live our lives today, the way we treat each other, the way we love, the way we laugh, the way we live. And above all else, future humans must respect one another's decisions and avoid unilaterally placing many others at risk. If you want to risk your own existence, that's on you. That's individual freedom. But you should not jeopardize the lives of everyone else. And the problem is technology could plausibly become that powerful. Again, I specialize in engineering evolutionary fitness, including engineering things that, if released in the wild, will spread. And we've seen what happens when you have an invasive exponential biological agent spread through humans lately. We know now that viruses can be more destructive than any nuclear warhead. And COVID-19 was not a particularly nasty pandemic on the scale of historical pandemics. It's been a horrific tragedy and source of loss for far too many. We could have done far more against it than we did. We made lots of mistakes. But our ancestors had it much, much, much worse. And even more, today, thousands of people can build a virus from scratch if they know what the genome sequence is. which leads me to wonder how we're going to manage in a world where not nuclear proliferation, but pandemic proliferation is a thing. Because there's too many of my colleagues who think that you can predict what the next natural pandemic will be. And they're brilliant, well-meaning people, but they don't think about misuse. They haven't thought that if you identify a virus that you think will cause a pandemic if introduced into humans, you've just given every researcher with the ability to assemble that virus and boot it up from synthetic DNA, the ability to ignite a new pandemic. That's just one example of bio. I am similarly more scared of things that can replicate exponentially than I am of most other technological threats. But it is why we need to focus on differential technology development. If we want to see humanity continue on our current trajectory of improvement, of lifting people out of poverty and suffering and anguish, giving them control over who they are and who they want to be, all of that is at risk if we discover the wrong technology that is sufficiently powerful, destructive, and accessible. So we need to learn to recognize when we're not ready to handle an advance, and if necessary, to tie our own hands. And so this is something that we're working on through a number of projects in which we hope to crowdsource threat identification. So anyone might have an idea of something that it would be horrifically destructive, but we don't want to share that information because not all of humanity is yet trustworthy. So what we need to do is crowdsource threat identification and build a computational system to limit access to the relevant synthetic DNA without disclosing what any of those threats are to anyone other than the people who thought of them. So that's the premise of one of the projects we're working on. And I hope it will be a template for what we might achieve in other domains of technology that might not be so simply encoded as biology is in DNA. That's all I've got. Looking forward to the discussion. Thank you.
Yeah, thank you, Kevin. That was kind of mind-blowing. We have some questions from the chat. The first from Gennady Stolerov. He is asking, what is your assessment of the relative ability of humans today to engineer more dangerous viruses versus engineering effective defense against viruses? Which set of capabilities is more advanced?
I think we are far better at engineering viruses than we are making defenses against them. And I think that's just been demonstrated by the most recent pandemic, because SARS-CoV-2, whatever its origin, was not intended to be a weapon. There's just no way. Anyone good enough to make it could have made something much worse. And what that means is that we're highly vulnerable to accidents. And as anyone in cybersecurity will tell you, any system vulnerable to accidents is fairly helpless against deliberate attack. Right now, we don't yet know how to reliably attack. We don't know which sequences will correspond to vast destruction. So my position is, let's not find out until we've got better defenses.
Fair enough. Fair enough. Thank you. We have another question from Robin Ahel. He's asking, besides making people immune to entire families of viruses, what more would be possible with the techniques you're using / applying?
Yeah, so I think of biotechnology right now as being driven by advances on 4 pillars. So one is PCR, making lots of copies of a nucleic acid, and that's what we use for our COVID tests. There's DNA sequencing, just reading off the code. DNA synthesis, making the code writing to whatever we want. And CRISPR for editing the code of what's already there. So any advances using CRISPR they unite the 3 major classes of information encoding biomolecules. That is DNA, RNA, and proteins. CRISPR is a protein that uses an RNA to find a corresponding DNA sequence. And so it's sort of the ultimate multi-purpose Swiss Army knife in biology. If you can get a CRISPR system into a cell and program it to, say, interact with other proteins and bring them to a particular DNA sequence, you can turn genes on, you can turn genes off, you can mess with the epigenetic state, you can label it so you can see it, you can direct things to different biological compartments. It's just a tremendously useful tool for all manner of engineering biology. So a lot of our work on multiplexing, just being able to target lots of things at once, would facilitate all manner of research. So the biggest application of CRISPR is has nothing to do with the clinic or even engineering animals or agriculture or wild species, editing mosquitoes to get rid of malaria. It's actually accelerating biotech in the lab. It's making all of the other advances happen faster.
Yeah, thank you. So we are kind of running out of time, so we will continue this discussion during the panel. And for now, let's have another break. Thank you very much, Kevin. We'll see you in about 15 minutes again.